Hi, this is Dr. Jabali, and in this video we're going to focus on topic 7.2, which is all about transcription and gene expression. Once again, this is an HL course. So what are the things that we're going to target in this, guys? This is where we're going to target the gene expression and how this gene expression will regulate certain type of proteins. We're going to talk about how the environment might affect some gene expressions. We're going to talk how the nucleosome might regulate some transcriptions. We're also going to talk how transcription happens from a 5 to 3 direction and how we can modify the mRNA to give different types of proteins. And by splicing it, we get these different types of proteins. We're also going to talk about the promoter, which is an example of a non-coding DNA, but it has its own function. And we're going to talk about the analysis of the change in DNA methylation patterns as we go on the ages. So I know you already learned this in topic 2.7, transcription translation. So I'm kind of doing like a quick review just to refresh your mind on this topic. But to start off all, if you want to make a protein, the recipe to make that protein is found all the way in DNA. And we know that DNA right here cannot leave the nucleus. And so that means you have to find a way of copying the information from the DNA. And once you copy the information from DNA, then you can take the information out to whatever place you need to go. So that copying, or should I say transcribing, is known as transcription. Transcription is basically making how can I copy the DNA. And in that process, you are creating mRNA. So mRNA is like a copy version of a section of the DNA with the information that you want. Once you make that mRNA, then the mRNA leaves the nucleus and goes to an area such, for example, it can go to the ribosome. And what does it do in the ribosome? The ribosome will create this protein right here. And this protein here is basically a translation that happened from the mRNA into protein, which is basically the reason why it's called translation. That's what the ribosome do. It changes the mRNA coding into amino acids. So with that in mind, let's talk about transcription. As it says, it's a synthesis of mRNA. Students think the mRNA already existed, that the mRNA goes to the DNA. That's not true. mRNA doesn't exist. mRNA is created from scratch once you enter the nucleus for the DNA template. So you are creating the mRNA by reading the template. And that means the DNA nucleotides will be copied into segments of RNA. And of course, the transcription will only be about a certain section. That means a section in which protein you want to make. But that section is known as the blueprint of the protein. So basically, DNA has the information to make all different types of protein. So DNA is a blueprint. So here, just to show you an example. So let's say I have here a DNA section. And in this DNA section, not the entire DNA will be transcribed. It will be transcribed in sections. So for example, maybe from here to here, it is one gene, and then from here is another gene, and then from here to here is a third gene. So right here, I have made gene 1, gene 2, and gene 3. These are 3 different genes, and of course they code for different things. So I'm going to pretend that the gene 1 here produces the eye, the gene 2 produces your hair, and the gene 3 produces actin, which is a protein found in your muscles. So this is what we're trying to show you, is that you're not going to transcribe the entire DNA. You're going to transcribe a section. Now, if you remember from transcription 2.7, you have an enzyme that works with this, which is known as RNA polymerase. Now, RNA polymerase, what does this enzyme do? He just goes around through the DNA searching for that area, for that gene that will give you what you want. So in this case, let's say it wants actin. So that RNA polymerase will start moving through the DNA looking for that gene. But how would you know that this is the gene? The only reason why he knows this is genes is because every gene, in the beginning of every gene right here, there is this black thing that I marked here. It's a section known as promoter. It's basically where it's more like an identification. So this promoter here is saying, hey, I am the gene that makes actin. So that's what RNA polymerase will do. It's like, oh, once I read this promoter right here, I know that this is the gene where I need to start. So not only do you have a starting, which is the promoter, but you also have the end, because you don't want to continue transcribing the entire DNA. That's so huge. We also have a terminator. And that terminator says, hey, this is where the gene stops. After this, you're not supposed to continue transcription. So every single gene has a promoter and a terminator. Okay? So in this case, the only ones that we are going to be working is this gene right here. So this is the one who's going to go through transcription and then eventually through translation to make the actin. So with that in mind, here is a more nice picture, better than my drawing, which shows again the promoter and the coding sequence and the terminator. So this entire section here, as you can see, is a gene. So the mRNA is actually going to start here and the mRNA is going to end here. Remember that this does not code for the mRNA and this does not code for the mRNA that we're going to create. It's right in the middle, the coding sequence. So here is a picture showing the white thing here is the entire RNA polymerase. And here you have the blue part, which is the DNA with its nucleotides. And the pink part here is the mRNA that is being created. As you can see, new nucleotides are adding right here as you read the DNA. Now, the new mRNA is going to be built from a 5 to a 3. It's very important that you know that this is going from a 5 to a 3. Always build up from a 5 to a 3, the mRNA. And of course, you're doing this by reading the DNA strand. Now, we also have to know which side of the strand. Is it this strand or is it this strand? Which one of these strands are the ones we're doing the transcription? So we're going to learn more about the sense and the antisense soon. But just as a recap, we saw that the only enzyme that works is RNA polymerase. Many students think that is the helicase that does the opening, and it doesn't. Helicase is only for DNA replication. When it comes to transcription, it is RNA polymerase that does the unwinding. It's the RNA polymerase that breaks the hydrogen bonds. And it's the RNA polymerase that reads the promoter region, which you can see here, the promoter, and then he starts adding the free RNA nucleosides into the templates, and it goes from 5 to 3 directions. So it's everything RNA polymerase does the whole thing, opens it, reads it, builds it, everything. And then, of course, when I say the word add nucleosides, it's very important that you know the difference between nucleosides and nucleotides. See, nucleosides, they lose 2 phosphates and as they bind. So nucleosides would have been like this. You have 3 phosphates and then the sugar and then the nitrogen base. Nucleotides will be 1 phosphate with the sugar and the nitrogen base. In order to bind, what they do is they lose these 2. And these 2, what do they give? They give energy to form the bonds. That's the difference between nucleosides and nucleotides. And of course, you're going to reach the terminator point, and then that's when you stop and you detach from the template, and now we have the mRNA. So you have created the mRNA, and then you release the mRNA from the DNA, right? And then the DNA goes back to be rewound. So you can see here, this section right here, it was the mRNA. And the mRNA, again, is a 5 to 3 prime. And I'm going to repeat this because it comes a lot in the multiple choice questions. It's always a 5 to 3 prime. So here is a very nice sum up for transcription. You have the initiation, the elongation, and termination. And you know what? If you mention these 3 stages, words, initiation, elongation, termination, you can get points in some of the mark schemes. So the initiation, RNA polymerase binds to the helix, and it starts to read and create the mRNA by adding RNA nucleotides. Elongation, you just continue to add nucleotides. Termination, transcription finish, the RNA detach from the terminator, the mRNA goes out, and DNA rewinds. So this is the step of transcription. This is a very good, great summary of what transcription is. Now we have to know about DNA sense and antisense, because this is HL course. This is the moment you have to figure out who is the mRNA being transcribed with, the sense or the antisense. Now the sense is the one that is always coding, right? And usually it looks exactly like the mRNA, except that the uracil is replaced by the thymine. Now on the DNA antisense strand, this is the plant that has the same base sequence as DNA, and in this case strand that is being transcribed or copied. So we're going to work on these right now, and I'm going to show you. Here is a picture showing the sense and the antisense. Now, I'm going to show you how to figure out these sense and antisense, because this kind of pictures are here you can easily find in the test. So I want to make sure to clear it overall. First of all, just to be clear, and I'll have here on the corner that the mRNA will transcribe, it will transcribe the antisense DNA. So it's very important, always. mRNA always transcribes the antisense. However, it is the sense strand that codes. And what I mean by codes, that means it is the sense that has the information. So the mRNA here needs to be a copy of the sense. So even though it's a copy of the sense, he will describe who? The antisense. Now let me show this in a drawing. So right here we have DNA strand, right? Say 3 to 5, and this is a 5 to 3, because you know DNA is a double helix, anti-parallel. And one more thing about the mRNA. mRNA is from a 5 to 3 prime. So 5 to 3 prime, that means it needs to be built somewhere here, right? Because it got to be the opposite. So 5 to 3 prime. Does it make sense? It cannot be in the bottom one because the bottom one is already 5 to 3. Now, I said the mRNA transcribes the antisense. So if here, as you see, it's created, here is the mRNA, right? So obviously this will be the antisense. And this will be the sense strand, right? Now, just to show you here with drawings, I'm going to show you this. I'm going to put the nucleotides for this. This is A, T, T, C, C, G, C, C, A, T. So what will be the opposite DNA will be T, A, G, G, C, G, G, T, A. So that was what I did. I just put the nucleotides in the DNA. What will be the mRNA? Now the mRNA, like I said, is transcribing the antisense. So A will be U, A, G, G, C, G, G, U, A. Now look very closely at the sequence of the mRNA. Doesn't that look a lot like the bottom one, the sense? The only difference is you can see here that the T's are actually the U's. But the mRNA looks exactly the copy of the sense strand. And that's the point. You want to make it an exactly copy. But if you went down here and created the mRNA here, right? What would the mRNA be? The mRNA would have been, with a T, it would have been A, it would have been U, and then a C, C, a G, a C, C, and this would have been an A, and this would have been a U. Now look at this. Would this right here, would this guy look a lot like the sense? No, it doesn't. So this is the reason why you do not copy the sense. You can't copy the sense because then the code will be completely different. It will look completely looking different from the sense. So just to be clear once again, the mRNA is built on who? On the antisense. So why? So they can look exactly like the sense. And mRNA is exactly a copy of the sense, except that you have the U instead of what? Of the T. That's it. And you will have a lot of multiple choice coming and tricking you with these answers. So let's play with this. I'm going to put it right here. And this is something that usually they will play with you. They will put here 2 things. And they will say, for example, let's say you have here A, T, C, 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 C. A, T, G, C, G, and then this will be T, A, C, sorry, G, 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 T, A, G, C, right? I did not put any numbers. And then they will ask you to create the mRNA, right? And the mRNA came out to be, for in this case, let's say U, U, and then here will be A, and there'll be G, 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 and then it'll be U, and then it'll be A, then it'll be G, and then it'll be C. Now this is the mRNA I have created, right? Now from this strand that I gave you right here, which one, number 1 or number 2, is the antisense? Obviously, you're going to figure out that the antisense will be the one that doesn't look like the mRNA. And if you look at this, number 2 looks a lot like mRNA, has all the GGs, the things, except that you see the U's are separate from the T's. So this definitely is the sense one, and this definitely must be the antisense. So number 1 is the antisense because, again, it's the opposite. And then the sense, which is number 2, looks a lot like the mRNA. This is one way that they can test you. Another way that they can test you is that they can just put the 2 lines here, and they can say this is a 3 to 5, and this is a 5 to 3, right? And they say, which one of these is the sense? And I'd be like, what the heck? Well, think about it. The mRNA, right? What is it made? Is it made from a 5 to 3? So that means it has to be here, right? Because it's building from a 5 to 3, and it has to be opposite to this. So if it's 5, 3, then this must be the antisense. And this must be what? The sense. So this one is the sense. And of course, the sense will have also the 5 to 3 just like it. So this is ways in which they can trick you in the test. After we have copied, basically I would say transcribed, not copy, I would say we have transcribed DNA, because when I say copy DNA, it's like replication. So keep copy DNA for replication, transcribe DNA for transcription. Now we have to go to the next process, which is known as the the mRNA processing. And this is the process where you're removing the introns. Introns, they are non-coding segments. If you remember from 7.1, introns, they do not code for proteins, and they are going to be spliced out. So what do we do? Basically, if you don't take them out, then you're not going to be able to produce that protein. So you must take the introns out. So here's a picture showing. Now, this is basically the DNA that has been transcribed. And when you see here in dark purple, I will say this is the exon regions. These are the ones that actually code for the protein. And the little pink ones, these ones are known as the non-coding. So where I'm going to highlight here in yellow the ones that don't code. These don't code, this don't code, this don't code, this don't code. So since they don't code, what do you need to do? You need to get rid of them. So that means all these yellows that came, all these pinks that came from DNA, these ones have to be cut. So by cutting them, cut them out, cut them out, cut them out. Of course, after transcription, right? What are going to be left? You're going to be left only with the purple part, which means you're only left with the exon. And that's when you made a mature mRNA. So immature mRNA will be where you have the introns and the exons. A mature RNA would be when you just have exons. That means the exons is the ones that actually code for it. So the cut is known as splice. So here's the different ways that you can manipulate the splicing. When I say the splicing, I mean is it cutting the introns. Here we have different exons, right? So alternative splicing will be like, oh, I just want to splice certain sections. So for instance, on the first protein A, what did I do? I spliced all of them, right? And I took all the pieces. So I took this, 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 and that. What did I splice? I spliced this, I spliced that, I spliced this. So the highlighted yellow were the introns that I took it off. And now look what I have. 1, 2, 3, 4, 5, and I have myself this protein. Now, let's look at the other one. On the second one, I just want 1, 2, 4, and 5. So I did cut this, I got this, I got this, and I got that. So what did I splice here? I spliced this entire thing, and this one, and this, and voila, I have this, and now I have a different type of protein. You can see how protein B is different than protein A. And then I can erase this one, right? Let's just get these all out. Let's go for the last one. Last one, I want 1, 2, 3, and 5. So I cut 1, 2, 3, and 5. These are the ones that I get. And I splice these, I splice these, I spliced all of this and that. And see, I have a different protein. So depending where you're splicing, you're going to create different types of proteins. And that means different mixtures of exons will give you different types of proteins, as you can see. A mixture of 1 through 5, you get this. A mixture of 1, 2, 4, and 5, you get that. A mixture of 1, 2, 3, and 5, you get that. So you get proteins. And that is just by splicing different sections and like choosing which introns you want to cut and which introns you don't want to cut and which exons you want to cut. So that's alternative splicing. Now, the most important part is that right here, this was just one mRNA. So from that one mRNA, you could create so many different proteins. Okay, that's very important to know. So after you have spliced, so let's say here you have the mRNA and you have the exons. Let's say this section is exon. Then you have the introns. Then you have the axon, an intron, and then the axon, right? I'm going to highlight them as yellow for the exons and green for the introns. So you have to go through the splicing. Splicing is basically cutting. So when I cut, I'm cutting who? I'm cutting the introns. So I'm left with only the exons. So this is the exons here. I have axon, the exon. Oh, actually it's supposed to be tinier. So it's like here, the axon, right? So that was what I was left with, is just the exons. Now, that's not enough. Of course, you got rid of the introns, which don't code for anything. The next step is that they have to add the cap. This is known as the cap, and then they have to add the tail. Now, the cap is usually made of meth, and the tail is made of a bunch of A's. That's why we call it the poly, because poly means many, and poly A tail. That's what he calls the tail. Poly A tail, because it has so many A's in the edge. Now, what does the cap and the tail do? These two, they kind of protect the information as they are about to leave the nucleus and go towards the ribosome, right? So after you do the splicing, you add a cap and a tail. Why am I saying this? If they talk about a question saying, explain the process of edifying the mRNA, talk about getting rid of the introns and keeping the axons, but right after this, you have to mention about adding the cap and the tail to each side of the axons, because this is what's going to protect. So, finishing with mRNA editing, let's talk a little bit more about the non-coding sections. Now, we already talked a little briefly about them in topic 7.1, but here in 7.2, we go a little bit more in details. See, the telomeres basically means the end. So it's like telo, for the word telo here, it means tail. So they are the end of the chromatids, and they are not transcribed. Now, what's the point of the telomeres? Well, the problem is that telomeres are like protecting our DNA. So what happens is, every time you replicate the DNA, you lose a little bit of the telomeres, but not the DNA coding. So as you get older, the smaller is the telomere. So like I said, it protects the chromosome from the loss of essential coding DNA and each cell division. So every time you do division, the telomeres become shorter and shorter. Let me show you here an example what I mean by that. So let's just say here is a DNA strand, and let's say from here to here, this is the telomere, all right? And the rest of this is DNA coding. Now, every time you go through cell division, that means you make a new cell, you have the DNA coding nice intact, but what happened to the telomere? It gets smaller. And then you go through the cell division again, and you can see the DNA is intact, but the telomere gets even smaller. And it will keep getting smaller and smaller and smaller. And they believe that this shrinking of the telomere, as you can see, it's decreasing every time you divide, is the reason why you age, is because the telomere gets smaller. So telomere is here for protecting the DNA content. Now, regulatory genes, this is another type of non-coding sections, and this has more to do with regulating the transcription. Translation means as you want to go forward or you want to stop it. So they're more like an activator or a repressor. Activator means go for it, repressor means stop. So let's talk about this gene expression, since we're talking about these so-far called regulatory genes. So here we have what we call as transcription factors, regulatory proteins, cultural elements. These are all different things here, like these ones that I'm highlighting here. These are all things that are participating in the gene expression. So one thing is important, as the RNA polymerase, see, it cannot initiate transcription at the promoter without what? Without the transcription factors. So basically, these factors are the ones who regulate gene expression. Without transcription factor, transcription doesn't happen. Now, we have regulatory proteins. They can be activating or they can stop repressing them. We have control elements, which they can be distal or they can be proximal. Distal means far away and proximal being very close. And of course, the distal elements, they can be enhancer or they can be silencer. Enhancer, that means go forward. Enhancer means stop. So all of these guys, they work on what? They work on transcription. They regulate if you want to go forward or you want to slow down, you want to speed up transcription. Now, the transcription factors, as we mentioned, who do they work with? The proximal elements. And the regulatory proteins, they work with the distal elements. So it's hard when you see a bunch of these names here, but this is just written. You can pause this with eventually knowing, but I'm going to show you this more on the next slide. You visually can see. But like I said, if you remember the proximals ones here, right? The proximal and the distal. Who are they? The proximal and the distals are the control elements. And then you have transcription factor and regulatory proteins, which are these guys here. These guys, they're very important for what? They're to regulate the transcription. Now, in the distal elements right here, right? The distal elements, they can enhance, right? Or they can be a silencer. Just like the regulatory proteins, they can activate or they can repress. So you can see the regulatory protein is more about go forward or reduce, while transcription factors is more like, am I going to start or not? Am I going to slow down or not? Here is a picture now. This maybe will help you understand more. So if you remember, I said that there is control elements such as the distal and the proximal, right? These are the control elements, as you can see. The proximal, you can see it's very close to the promoter, which has a Tata box, and this is where the RNA polymerase would start the transcription here, or the DNA sequence, right? Here's the DNA. RNA polymerase does the transcription. And remember how the RNA polymerase attaches the promoter? Right here. So next to the promoter, you have the proximal elements, and far away, you got the distal one. So these are the control elements. Now, who touches the proximal? The transcription factors. Who touches the distals? The regulatory proteins. Now, the regulatory proteins, there's two types. There's the activators, which this one will increase the transcription, and there's the silencers, which decrease. So if the regulatory protein comes in here and touches the distal E, which is the distal element, but it comes as an activator, then voila, transcription will increase. If it does the opposite, right? If it doesn't come, if the regulatory protein comes here as a silencer, then what happens? It would tell the distal no transcription, so it decreases, and that means they will block the transcription from entering. So let me repeat this part very clearly so you can understand. The transcription factors, these guys, they will only touch the proximal with the command of these guys. Okay, so if the activator is the regulatory and it touches the distals, that means the transcription will touch the proximal element. However, if it is the silencer that is going to be the one that it touches the distals, then it will not allow the transcription factor to touch the proximal. So that means no transcription happens. So you can see that the regulatory proteins are the ones who control the increasing or the decreasing of the transcription. But again, they have to touch a certain area. What exactly is the area? Is the control elements. They have to touch the control elements. So what do you need to know? You need to know the control elements, the distal and the proximal. You have to know that it's only the regulatory that bind to the distals and transcription binds to the proximal. And the regulatories, you have two types. So if you know this, then it's easier for you to write an essay on it. So here's a picture that I found of BioNinja, which is pretty nice because it shows a little bit more fancy drawing than my drawing, but maybe now you can understand what this means. So here highlight, you see this is the DNA, right? And you can see here the DNA. Now you can see here is an enhancer, and on this side is a silencer. So this enhancer here is what? The regulatory protein. Who's it touching? He's touching the distal right here. And then here we have, of course, the proximal elements, right? The proximal. And here you have the transcription factors. and they touching the proximal. So you can see the transcription factors is touching the proximal while the enhancer is touching the distal. And this leads to what? To the RNA polymerase here to go ahead and do transcription. On the other side, you have the silencer, which is the regular protein, touching the distal, right? And this will cause this part here, which is the proximal element and the transcription factor, they will not be working. And this way, what do? You stop the transcription. So I don't know, I prefer my drawing here, but if you like the other one, it's totally fine. But this one, I think it gives more of a process of understanding how it works. But you have to know where do they touch, where they're located, and what's their purpose. So the next thing we're going to talk, it's about how we have the external or the internal environment affecting the gene expression. So depending on what the environment is saying, the gene would express into something. So let me give you examples what I mean by that. So we have the Hydrangea. What is this? This is a type of flower. And basically that flower, its color change according to the pH of the soil. So for example, if the soil is acidic, this plant will produce a blue flower. If the soil is alkaline, it will produce a pink flower. So you can see that depending on the pH of the soil, the gene will express a different color. Another example is a Himalayan rabbit. Now, the Himalaya has the same gene. And depending on the temperature, you will produce different types of fur. So for example, if the temperature is above 35 degrees, it will produce a white fur. And if the temperature is below 30, it will produce a black fur. This is, again, the gene is depending on the environment. Humans, believe it or not, humans produce the amount of the melanin, which from the skin pigment, and that's dependent on the light. Too much light, more melanin. Less light, less melanin. So you can see they all influence. And this can be also with fish, reptiles, and amphibians. They all change their gender according to social cues. Or for example, we know the reptiles, for instance, if the temperature is too hot, you become a female, and if it's too cold, they will be a male. So the environment can change the way the gene is expressing. So the genes are not fixed. The genes can be manipulated if you change the environment. So here is just showing you an example. Like here we have a stimuli, right? If the stimuli, as we saw it, if it's a pH of 4.5, it will give me a deep blue. If it's a pH of 6.5, it will give you a medium pink. If it's a pH of 7, it will give a deep pink. So you can see it's the same gene. You're using exactly the same gene, but depending on the stimuli, depending on the environment, it will give you a different result. So there's no such thing as gene just deep pink. A gene can produce different types of color. It just depends who's going to touch the gene. Next, let's talk about how we are regulating the transcription and nucleosomes, right? Of course, with nucleosomes. Now, if you learn this from 7.1, the tighter the nucleosomes, the less transcription, the more loose the nucleosomes, the more transcription will happen. So remember that we have this histone tails right here, right? And they have a positive charge and the DNA has a negative charge. So that's why they're so very much tight. Now, adding acetyl group to the tail, which is acylation, what does it do? It basically, right here, let me highlight it, it neutralizes the charge, making the DNA less tightly coiled and increasing transcription. Very, very important to read this. Just to read it again, just to make sure you understood. Again, if you add acetyl group, which is known as acylation, it neutralizes the charge. That means the positive and the negative. And of course, DNA being the negative and the histone being positive, this makes it neutral, right? Because the plus and the minus, they cancel each other, become a zero. This makes the DNA less tightly, and that means increases transcription. So what does this mean? Acylation right here does what? Increase transcription. On the other hand, if you add a methyl group, right? This maintains a positive charge, making the DNA more coiled and reducing transcription. So methylation right here reduces transcription. And it only has to do with the charges. More positive would cause less transcription. More neutral, more transcription. So here, let's put it here. If it's neutral, that means more T as in transcription. If it's positive, less transcription. If you can remember this in your head, it works. Also, we can say that the acetyl increases transcription and methyl decreases transcription. So having this cheat sheet right here, it will help you a lot. Again, acetyl makes it more neutral, more transcription, and you can also say loosen up. Methyl makes it more positive, that means less transcription. That's very important. It can be a multiple choice easily. Here is basically showing again. So the acetyl, as you can see, what is it does? It increases the transcriptional by doing it, making it more loose. You can see how they're more loosened up. There's a space between them, right? So acetyl makes it more loosened. On the methyl, on the other hand, you can see they're more tied to each other. There's less space between them, and this means no transcription. So it makes sense that the more they're loose, the more transcription will happen, and the more they are tight, the less or no transcription will happen. That's the whole point. And then let's go for the next thing. Since we talk about methylation, which the more methylation, the less transcription, let's talk about how the methylation of DNA affects gene expression. Now, if you increase methylation of DNA, it decreases gene expression. We already got this point, right? Because more methyl, less transcription happening. Genes are not described tend to exhibit more DNA methylation. That's obvious. We already got this. More methylation, less transcription. Now, life point DNA methylation factors. So fertilization, for example, pregnancy, infancy, adulthood, senior. These are ways in which it stops gene transcription. So for example, in fertilization, it might be just that the genes are not allowing. Pregnancy, whatever the mother is eating. Infancy, how much it's being exposed to the microbes. Young adult, the environment, the diet, what they're doing, and age-related changes. So these are saying that these are sections in your life in which methylation is affecting your body. So from these data, what did they figure out? Okay, before I even get there, let's first define what's epigenetics. So epigenetic is a study of changes in the phenotype as a result of variation in gene expression. So you're trying to see how the trait will look from the outside by seeing how the gene inside is expressed. So basically, epigenetic is just studying the gene expression. Now, what the analysis have shown, that DNA methylation patterns may change over the course of lifetime. Like I said, here, this through lifetime, each section, what did they do through the diet, through the exposure to microbes, through the environment, or to the age-relators, they change the methylation. It is not genetically determined. Why does that mean? Because they done a test on identical twins and they show that they have different DNA methylation patterns. So it's not a gene that says, hey, you're stuck with this number. Okay, so it's not fixed. That's what I mean by predetermined. It's not fixed. The genes do not fix the amount of methylation you have. Different cell types in the same organism may have different methylation patterns. So the amount of methylation in the lungs might be different than the amount of methylations in your heart. Also, you have the environment, diet, bacteria, etc. They may influence the level of methylation in the cells. So your environment can make you increase methylation, or your environment can make you reduce the methylations. So here there's two studies. One study was just talking about comparing the methylation patterns of identical twins, right? When they were 3 years old, and then later on when they were 50. 3 years old when twins to 50. What the finding? The methylation patterns differ between clones and changed over time. Methylation patterns have a genetic basis but are influenced by the environment. So basically, in the beginning, you can say they might have some differences, but it gets even bigger, the difference, as they get exposed to the environment differently. Here, the comparison of methylation patterns in healthy and diseased individuals of twins. The methylation patterns differ between the healthy and the unhealthy ones. Methylation controls transcription and hence phenotypic expressions of the disease. So you can see even when one has disease and the other one doesn't, there's a large change in the concentration of methyl. So this is just proving one thing. Methyl can be influenced by the environment, and it's not predetermined. That means you're not burned with a certain amount of methyl, because they used two twins and they were expecting the two twins, since they have the same gene, they should have the same methyl. It's not true. The twins came out with different methyl, right? Amounts since they were born, and one had healthy and one had disease. What happened? It changed even more the difference in their methyl. So these is both case studies are just proving that methyl is controlled by the environment and it can change throughout your life, and it's not determined by a gene. And that's pretty much it for 7.2. Here is the study guide for this section. All the questions can be answered in this video, and if you know how to answer them, then you have mastered this topic 7.2. Thank you.